In this story, we look through a virtual microscope to learn how visualization can improve our understanding of the impossibly small world of cells, such as this human-induced pluripotent stem cell. But how is a virtual model of a cell even created? Our journey begins in the lab, where scientists capture images of living cells using 3D fluorescence microscopy. Many functions of the cell are carried out by large structures called organelles. Organelles are so small and densely packed that it is difficult to discriminate them from each other when using a light microscope. Scientists derive light-emitting proteins from fluorescing jellyfish to light up just a few organelle systems at a time with different colors, in order to improve their ability to distinguish them from each other. The part of the jellyfish DNA responsible for producing the so-called green fluorescent protein is transferred to the cell. The cell will start to produce the protein on its own, which will cause the tagged organelles to emit colored light when excited with a laser. A spinning disc confocal microscope is able to focus a laser beam into a very thin layer of the specimen to stimulate the tagged organelles to emit light. By continuously changing the focal plane, the microscope scans the specimen layer by layer. The result of this process is a stack of images that form a 3D volume representing the structure of each organelle system. This process is repeated for each type of organelle: the microtubulus, the mitochondria, the nucleus, and the cell membrane. When put together, the volumes do not convey much, so the question is, how can we improve the representation of our data? We start by blurring the surface of the membrane to smooth out the rough edges. Then, we extract the surface and we repeat this procedure for the nucleus. Now, we can display both extracted surfaces together. The smaller organelles are still fuzzy, but we cannot extract the surfaces by blurring since they are too fine. Instead, we use a segmentation algorithm to identify the individual mitochondria. We display them with so-called metaballs, blobby geometrical objects that nicely approximate the organic shapes of the mitochondria surfaces. Even though the mitochondria now appear as smooth three-dimensional objects, they're actually represented by two-dimensional textures. These so-called billboards are always oriented towards the camera. By using this technique, we save a substantial amount of processing power. The microtubulus are also represented with this technique. However, since they are 12 to 40 times thinner than mitochondria, we have to give them a much smaller radius. Now we add some depth cues to support the understanding of the complex 3D structure. And voila, our virtual cell is born. Our story ends here, but the journey of our cell has just begun. We are continuously working on expanding its internal architecture by adding new organelles and organic components. Soon our cell will find that all of its parts are composed of millions of proteins and other molecular structures, but that's a story for another day.